So welcome to Symposium 31, which is Stronger Together, updates from the International Clinical Trials Network, actually, because we're not going to present any clinical trials data. We're going to be talking about the work that has been done across international networks, and it ties in nicely to some of the things that were highlighted by Dr. Liz Tullis at the last plenary. So we really are looking at giving some nice updates and summaries on the international collaborations, as noted in the slide, because the existence of the networks that have been in place for quite a while gives us all the opportunity to think about ways to truly expand the population and gather samples across the existing networks. And let's see, so our objectives are going to be to look at the approaches used to design protocols that will allow sharing of biospecimens and data, and also then to look at developing consensus around the elements for a robust and rich patient registries, and finally to address some of the issues around global standardization in the development and dissemination of biomarkers that are essential to the development of new therapies and have been a key part of what some of the networks have been working on for quite some time. I wanted to give a little bit of a perspective on the networks, and my own perspective comes from being part of the Cystic Fibrosis Foundation's Therapeutics Development Network for the last 24 years. And so networks have been a successful feature of collaboration, and so here in this map, you can see in blue the sites across the United States that are the Therapeutics Development Centers. And then soon after that, the Success with Therapies Research Consortium, which is looking at network collaboration from a very different perspective, and also a consortium of sites that are very interested in assessing therapies and outcomes for non-tuberculous mycobacterial infection, the CF Learning Network, which is really helping clinical centers develop improvement in the delivery of care, and the most recent consortium that focusing on lung transplantation. So that's the United States. In 2008, I had the great pleasure of getting to know a growing European CF clinical trials network that is now grown to 57 CF centers in Europe, spanning 17 countries, over 20,000 adult and pediatric CF patients, and incorporating the strengths of four very large national networks in the UK, France, Germany, and the Netherlands. And so this is one of the networks that reflects the extensive collaboration with the TDN, as well as in the UK and Canada. So the UK program is fondly known as CTAP, which is a Clinical Trials Accelerator program that was established in 2016. 18 CTAP centers that have a nice regional pattern that we are trying to model in the United States with affiliate centers to really enhance the opportunity for people with CF to engage in clinical research, even if their own center is not a major research center. This has been a really very busy and very productive network, and includes a number of very experienced early phase programs. The Canadian Clinical Trials Network, or Accelerating Clinical Trials, CANACT, was founded in 2018 and expanded 2 years ago, and this map and the list shows the wide span of centers that are participating in that network. We are hoping to hear from the Australian and New Zealand groups in their discussions that they've had with us in the past about establishing a network as well. So truly a great opportunity to work across the globe in areas that have the greatest interest in populations for CF clinical research. So today we're going to hear about four very different but highly important programs that are going to be looking at, are presenting the approach to COVID antibody responses, to individualized therapies in CF, global harmonization of registries, and finally standardization of biomarkers. And we're going to try to make it less of a distant approach for questions so that we can do a hybrid approach to questions. And after each speaker, we can have a microphone and you can come up and ask a question live, and we won't have to paraphrase it. So I'm going to introduce my co-chair, Dr. Jonathan Raymond from University of British Columbia, who will be introducing the speakers and fielding some of the questions along with me.
Great, thanks, George. So, oh yes, the clicker, very, very important. So our first speaker is going to be Damien Downey from Queen's University in Belfast, who's going to talk to us about, very sorry, too tall. Our first speaker is going to be Damien Downey from Queen's University of Belfast, who's going to talk to us about the COVID antibody response study, which is sort of an exemplar of one of these multi-center studies leveraging the infrastructure within the clinical trials networks. So.
Okay, well, thank you for the introduction and the very kind invitation to speak today. So, as mentioned, I'm going to talk a little bit about the processes of how we started this study and how it is progressing. So, only a few disclosures relating to this trial or study from the CF Foundation for funding, and I'm director of the CTN. So George has shown how we have moved from the inception of the CTN, which was around 2008, and the whole concept of that at that stage was to bring research centres together to help develop generally pharma-sponsored clinical trials to bring new medicines to people with CF. There has been various expansions over the years, and you can see our last expansion, we are now Yep. Are we good? Nope. Can you hear me?
I guess it wasn't fixed. I guess we could always just give you like
Test. Test.
Hello? Nope. We can bring people closer and I can just shout.
Okay, in the meantime, if people want to come a little bit closer, we can just shout, because I'm not sure if we're actually going to be able to fix the AV in time.
This was planned?
Yeah, to get everyone sitting closer together. Oh, okay.
It did, yeah.
I was hoping it would be fixed. And I want to reassure you all, this is not an example of American censorship.
Just when I was talking about expansion of the CTN.
Okay.
Can you hear me? Oh, that was encouraging.
It's really just
Dead.
press on.
Yeah. Well, let's continue, and hopefully you can hear me without microphone. So we're currently 57 sites, and we've moved from about 9 sites in 2008, and our last expansion has gone up to now 57 sites in 17 countries, which I think is a fantastic achievement. So a little bit about the work that we do. This was initially, as mentioned, designed to get a platform for pharma-sponsored trials, and you can see the active CTN studies over time from 2009, obviously a lowly 1 study, and then a migration over time to, this is data up to June of this year showing there's 25, 26 active studies within the CTN. We do produce a report which you can get online from from the CTN website. So just search for ECFS CTN on Google. You will get our CTN website, and you can download our report. And this is just some of the data on the left-hand side of 2021 in numbers, our publications, and so forth. And we will publish a lot of our protocols for standardization, which Isabel, I guess, will touch on later, and some of the protocol, the last protocol will be on nasal cell data that we pull on how to do that across Europe. You can see there about the protocols that we've reviewed, the feasibility checks on the studies, and some of the early data for CAR-CF. On your right is an overview of what's happened to the CTN in the last 12 years, and you can see there we have reviewed over 172 protocols, 100 feasibility checks, and we have supported 112 clinical trials in the European network. This is our structure. We are a project of the European CF Society. We have an executive committee, which I'm director of at this time. There's a co-director, and then other PIs, pediatricians, adult CF physicians, and a patient representative for CF Europe, which is overseer of the patient organizations across Europe. We have a coordinating centre based in Leuven in Belgium, and then we have a steering committee, the SteerCo, which meets in January and in June, where all the investigators, PIs across the CTN meet, where we plan the strategic moves that we do within the CTN for the coming years. Now, what's really important are our various committees that we have, and you can see just at the bottom there's a protocol review committee, which was our main committee for reviewing pharma and sponsored protocols. We usually have 5 to 6 different groups, so we can look at multiple protocols at the same time. Standardization committee headed by Isabel Sermet, which you will talk about, and a training committee for our research coordinators. The newest kid on the block is the investigator trial committee, and this was started a couple of years ago in conjunction with starting the CAR-CF study. So they morphed together, and I'll march on to talk about that now. So what about CAR-CF? This was a study that was initially born within the Canadian network. They contacted us and said, would you be interested in maybe engaging some sites? And I thought, well, we should really take this as an investigator-led trial across our European sites. And it was also in conjunction with the TDN. So this was quite a first for us as an investigator-led trial, coming across 3 networks. And of course, doing this at the start of the pandemic, trying to address a question about the pandemic, was again quite an unusual challenge for us all. So the initial question, and you may have seen a couple of years ago some initial studies looking at seroprevalence within 1 or 2 countries. We wanted to do this across the large network. And of course, that question becomes of less interest over time because the seroprevalence, one would expect the vast majority of people to have been infected with SARS-CoV-2. But things have changed, and I'll explain that in a little bit. The importance then is of sharing a core data set across those sites. The protocols, whilst we are very similar in our approach, we developed a European-specific protocol, and we took that within our investigator trial committee and wrote the protocol pretty quick. At this time, the vaccine was being developed and rolled out, so there was a further change, and it was a very iterative process. So as soon as you were writing something, new science came along, vaccines changed, it had to be rewritten, and that was just the nature of the beast coming at this time. This protocol was then reviewed within our internal processes, and then we moved on to putting this protocol into a grant application. This was led by certainly the heavy lifting was done by our Canadian colleagues, and we are very thankful for that. But of course, it was very much a team effect here from Europe, the US, and Canada, and we're very grateful for an out-of-cycle application to the CFF. So let's talk just a little bit about CAR-CF. This is a summary, this is it on a slide, and we tried to make this as inclusive as possible. So it was people with CF of any age, genotype, transplant status, disease severity across sites. So if you had a confirmed diagnosis of CF, then you were eligible. Importantly, the seroprevalence is just not a single point. It is a longitudinal prospective cohort study, and this is serial sampling of those individuals. Conducted over a 3-year period, so this is a 1-year enrollment period for each site, and then a 2-year follow-up of those samples. And of course, the initial question, as mentioned, was the seroprevalence in an international CF cohort. But it was also now we would be able to look at seropositivity over time, associate that with clinical symptoms, clinical outcomes, and then being able to also to compare antibody levels and those who are vaccinated, natural infection, maintenance of those antibodies, attrition of those antibodies, and to see whether this behaves differently in a CF population. So we can cut the data in many ways. The data collection is the basic demographic data and clinical symptoms and any infection, COVID-related infection details, and I'll talk a little bit about that. So the blood samples are collected on a 6-monthly basis. Of course, we thought about should we mandate this, and the challenge at this time was that not all patients were coming into clinic. So we didn't want to have a whole range of protocol deviations. So we had suggested to sites, do your best at getting whatever samples that you can. If there's routine clinical reviews, try and take a blood sample. Also with that, if people with CF were coming up to clinic for another sample, such as a liver function test because they were on a modulator, then we would treat that as opportunistically and take a sample at that time as well. We would are having a central laboratory in my university in one of our commercial academic labs, and this will be for measurement of the antibodies. The testing platform, and we are having ongoing conversations. We're trying to align the same testing platform across North America and Europe, but there's different platforms in each jurisdiction, so that will take a little work. And of course, within this as well, we have changed, we've had additional studies for additional consent where we will be able to do other studies on those samples related to COVID. So the clinical data, I think is important to discuss. This is from healthcare records and recorded on case report forms. This is a little bit different from our colleagues in Canada who are using the registry. European registry collates basic data from different types of registry across Europe, and of course we wanted a lot more detail, and we wanted to be live detail about vaccinations, the type of vaccination, the date of the vaccination, and so forth. So we thought we would create our own case report form across countries, and we now have an electronic CRF based on a REDCap system where we can just pull all the data, which is working really well. So what about the structure? And this is slightly unusual. This is a consortium of countries, so we do not have a central CRO based at that time on costs and speed. We thought, how are we going to do this study with essentially limited resource? So we thought about doing individual CTN country leads. So it's run in individual countries, and then we pull the data at the end. We need to have a country lead, so we had a nominated PI from each of the country that would oversee the needs of that country for the study. And that is quite a lot of work, and we're conscious that we have asked a lot of our colleagues across Europe because the site and country-specific requirements can be quite challenging to do and are very there's a lot of time challenges with that. Sponsorship, and I want to talk a little bit about that. Obviously, a sponsor being the legal entity for a clinical study or a clinical trial, and that would be within each country. We had talked about a university hospital, for example, doing that across Europe, which can be done. However, that requires negotiation for insurance and contracting with each of the sites across Europe. And that is a lot of work, whereas if this is devolved to individual countries, it becomes quicker. Ethics as well, for example, in the UK, that's across the whole of the UK, it's one application. Different countries have got different ways of doing ethical. The board may be just for that site, it may be for the country, and in some countries it can be a combination of individual sites and a country specific. That's obviously changing with European regulation currently. And then what about the local costs? Now, certainly most of the costs that we have from the CFF were for antibody testing, transport of samples, but there's a lot of local costs and finances required as well. So we have asked sites to try and source what they can. We have a study management group, and finally we have a monthly meeting of our country leads, and we have an online repository for a protocol, lab manual, and all the study-related documents. So, briefly, this is from our CTN website, and you can again get up-to-date information about the trial on the project. You're not clearly meant to read this, but this is the participating sites within the CTN, and you can see the breadth of sites and countries, and this is a really fantastic resource because then you have such a wide variation across our continent. There is a huge support network for this. The CTN Central Coordinating Team have been pivotal in pulling this together along with me. We have our Investigator Trial Committee. We are again very thankful for CANACT and our relationship with them and the TDN. My own university for doing some contracting, and I will talk a little bit about that. And then CFF for funding. And the patient organisations have been really helpful in trying to source additional funding. But of course, if you're doing a study across Europe, all documentation has to be translated into all the European languages, and they have been critical in doing that and helping that with us, or for us. We have a study management group, and lastly but not least, is the various country leads that have been overseeing this devolved clinical trial in their respective country. So what about the challenges? Financing we have talked about from CFF, and we have country-specific patient organizations. Sponsorship is really quite difficult, as I've mentioned. We are doing that on an individual country basis. Contracting. We wanted to make contracting as simplistic as possible, and therefore the only contracting we have across sites are our material transfer and data sharing agreements. So in our university, we reduced quite a skeletal form. We took out a lot of the legal jargon to make it as simple and as basic as possible, and thinking this would be the way forward. I was clearly very wrong in that, because when it went to all the individual sites, all the legal country-specific jargon went back in, and then it had to be lots of negotiation between each of the countries. We kind of know how to do that now, but that has been a real challenge. Talked a little bit about the ethical committees and the board applications, and of course the translation of those documents. We haven't, again, the challenge you see with the advancing and very dynamic science of our antibody responses, and we're going to align our European and North American protocols for this. And I think it's important that the individual sites also have their challenges, competing studies, and of course you bring in an investigator-led trial, which may not bring in the finances that one would get with a pharmaceutical-sponsored trial, then that can maybe push those really important investigator-led trials a little bit back in the line, and therefore startup can be slower. Staffing levels and the financial support, because we have got central funding to run this study, but there's lots of individual charges, so PI time, research coordinator time, lab time, and so forth in each of those sites. Communication is key. We have several newsletters. We send all our data to all our sites. We update from our Canadian colleagues and US colleagues as well. I'd mentioned the annual report, and I think this is a really great resource, and this is a shameless plug, of course. And within that report, you will see a patient talk about their involvement and the importance of doing these large studies across a network and being part of something bigger. And as mentioned here, is a huge European study and requiring this unification in these difficult times. And this is really an important aspect of our study. On our website, you will see an update of where we are with the study. And there's a map that will show the number of people with CF that are recruited at this time. And this is our latest number that we have. I mean, we have over 1,200 people with CF recruited into this study. I think this is quite a remarkable achievement. And we would hope, just from a European perspective, have around 1,500 to 2,000 patients within this study. So where do we go from here? How do we do these studies? I've just used one example of how CAR-CF as an exemplar, but the CTN can be certainly used as a platform for investigator-led studies. We're very keen to pursue this. We want to diversify our portfolio. Of course, we maintain our pharmaceutical-sponsored trials, but having investigator-led trials are really important, because then we can answer those questions that may not be of interest to our industry partners. There are many platforms at this time, James Lind Alliance and so forth, to look at research priorities. So we can look at those important questions, as mentioned, that may be less interest to our industry, drug A versus drug B, stoppage trials, and so forth. And then bringing together our research networks. This is just not CTN, but CANACT and TDN. And with our, we're trying to get design teams together. And this is looking at encounter-based studies, decentralized studies, cohort comparisons, and looking at those novel designs that Nicole discussed on the second plenary as well. And of course, the stoppage trials and the really important information that Simplify has provided at this time. Our networks are a perfect way to do those types of studies where you need large numbers. And of course, as some of the endpoints that may become more challenging in CF, where we may need larger cohorts to see smaller changes, then networks are the way to do this. And of course, as always, financing. So, summary. CAR-CF is a large European and North American study involving those networks. We are exploring longitudinal antibodies to SARS-CoV-2 and vaccination in people with CF. I hope I've showed you some of the challenges to starting an investigator-led trial across multiple countries and multiple sites. And we have developed a European consortium for this. This may not be the process for our next investigator-led study. But it has been a relatively novel, quite agile, multi-country investigator-led study. Recruitment is ongoing. I say we're well over 1,000 patients. We hope to get to close to 2,000 patients. And there's lots of opportunities for future studies. This is my final and completely shameless plug for the CTN, that we are open for business and very happy to have any discussions. Thank you very much.
Thank you for the great talk, and the microphones are back on just in time. I don't think there are any questions online yet, but I can walk down with the microphone. If anyone has any questions, you can meet me at the front here.
So, Damien, just one question about very time-sensitive startup. I think that at the beginning, when you proposed really looking at the response to SARS-CoV-2 and serology and clinical things, so you didn't have an idea that it was going to be such a long pandemic with a broad opportunity for enrollment. As I was thinking about the rollout of modulators and how you have a very short timeline, do you have a sense of, for future plans, given all the things that you learned with this, how long you think startup would be if there was another time-sensitive opportunity where you wanted to capture data in baseline and then after that starting point?
That's a great question, George, and we have learned an awful lot, and we are starting looking at, I guess one of the challenges was CRO and having an appropriate CRO. We decided to do this ourselves, so we're pulling the data of what's available, what CROs can work with us and costs and so forth, so we should be good to go with that type of study again if it was a time-sensitive issue. We know and the importance of the MTAs and DSAs, and I thought that may have been a slightly more straightforward process than it was. We now know what the requirements of each country specifically, so we would be able to be much faster with that. I think the big challenge was financing, and we're very grateful for CFF, but it was a restricted amount of money that we could apply for, and a study like this would take 5, 6, 7, 8 million euro with absolutely, because there was costs each site needed, and we asked them to try and finance that themselves, which again delayed that. And back to my point of a study, an investigator-led study, may be pushed a little behind other more profitable pharma and just pharma-sponsored studies. So we know all this now, and we will have processes to deal with it. So if it was time sensitive, the next one we will be quicker.
Yeah, it was really impressive, the list of barriers that you had to overcome and the painful lessons learned from that.
No questions from the audience? Okay.
I'm excited to see that you have plans for future trials, because I think one of the other points is that networks will have to undertake certain kinds of studies that are absolutely vital to answer for the community based on the feedback we get from our people with CF in the community, and industry sponsors are simply not going to be there. So this is something that we will have the opportunity to continue to refine and collaborate on.
Yeah. No, we welcome the collaboration, absolutely.
I've got one more question. Can you talk a little bit about how the investigator-initiated study subcommittee is sort of evolving with the lessons learned from going through CAR-CF? Because taking on unfunded studies is obviously not something that you can do all day long.
No, we can't do that, and the challenge with that is that it was a big ask for our investigators to do, particularly the country leads, to take on a lot of extra work, so what our recent discussions within our investigator trial committee is just that: what will be the next project? We are trying to put some ideas together, and then for our next steering committee, which will be in January, then we will have a conversation with the whole CTN PIs to see how we proceed with the next one. So it will some ideas will be born out of that meeting.
Great. Fantastic. Thank you so much for the talk.
Thanks.
Cheers. Switch. So, thank you, Damien. Moving on to personalized CFTR modulation, I'd like to introduce Cors van den Ant from Utrecht to talk about the European HIT CF project.
Well, Jonathan and George, thank you very much for inviting me to talk about this. I'm happy that the microphone is still working, and I hope it will continue. And much respect for you, Damien, to have a lecture in this huge audience for so many people without a microphone, so I'll never forget it. So this session is about collaborations and how to move forward together. And I think that the HIT-CF project is really an example of collaboration. It's originated within the clinical trial network of the ECFS as an investigator-initiated project. And it's collaboration between many centers, but also a collaboration between patients and caregivers. And it's also a collaboration with pharmaceutical companies, both within Europe and within the US. So there's collaboration all over in this project, and we have learned a lot over time. Well, the goal of the project was to, let's see. These are my disclosures. Okay, then I can skip this after reading. The starting point was the ultra-rare mutations that do not have any access to CFTR modulating therapy until now. And well, we have talked a lot about it during this conference, that the new compounds coming from the Vertex company, like ivacaftor, lumacaftor, tezacaftor, and elexacaftor, and all sorts of combinations, that they cover up to 90% of the patient population, and they can be treated now. And we were worried about the last 10% of patients with very ultra-rare mutations that were not covered by this type of treatment. And they are patients with stop mutations, with difficult splice mutations, with frame shift mutations, and all sort of rare, sometimes uncharacterized mutations. So we were wondering whether we could help them and bring treatments to this last remaining group of patients. So we started the project in January 2018, so almost 5 years ago already. And the plan was to have a 5 years program, but well, due to many intervening things like the COVID pandemic and changes in the biotech industry, we didn't succeed to end the project within 5 years. So it's still running, and we have asked for an extension. It's funded by the European Commission, and they approved an extension for 2 years until now, of course, without extra money. So we have to solve these type of problems in these collaborative networks, as Damien already addressed. So the goal of this project was to get drugs available for CF patients with ultra-rare genotypes. And the second goal was to further validate the organoid model as a predictive tool. And you have already heard about the organoid model many times during the last years, and also during this conference. There was a lot of attention towards this model that can be used as a sort of predictor of therapy response in vitro for in vivo clinical responses. And the third goal of the initiative was to create a biobank that is available for the CF community now and in the future. Well, this is how the consortium looks like. And you can see on the top already the ECFS CTN, and Damien was talking about it, but also CF Europe is a partner in this project. It's a full partner, and they are attending all the meetings, and they are discussing along with the caregivers and the researchers, and they are there as an umbrella organization of all patient organizations within Europe. There are some laboratories, academic partners in the consortium from Utrecht, from Lisbon, from Leuven, are in the consortium, and these laboratories are working on the testing of the organoids. And several pharmaceutical partners are still in the consortium, and today there is Elox in the consortium and Fair Therapeutics. And it was really a sort of roller coaster of biotech pharmacies, because when we wrote the application for the European Commission, well, some 6 years ago, in the first round there was Nivalis and Galapagos was available together with Flatley Discovery Labs at that time. But between the first and the second round of the application, already one of these partners went down, and Nivalis stopped their development in the CF field. So we had at that time only 2 partners that were Galapagos and Flatley Discovery Labs. Well, as you know, Galapagos was taken over by AbbVie, or the CF program was taken over by AbbVie, and Flatley Discovery Labs also stopped its development when the effective modulator therapy from Vertex came up. So there were a lot of changes, and we had to discuss with new partners in the pharmaceutical company during the project. So it was really a challenge to keep the project running. And well, in the end, we could collaborate with Proteostasis. They had a nice triple combo of CFTR modulators, but Proteostasis was in a merger with Humanity, and Humanity was not interested in the CF portfolio, but only in the NASDAQ registration. So they stopped the development of the CF project, and we had to, well, to keep up this opportunity for the patients. And so a new company came up called Fair Therapeutics, and they now have the license for the former Proteostasis compound. So they are still in the project together with the compounds of Elox. There are also some supporting partners. Julius Clinical is a CRO company, and Biotech Subsidy is a partner that helped us to write the project, but also to communicate with the European Commission. And I don't know how it works in the US, but the Europeans are very helpful to make everything very, very complicated when it goes about applications. The website alone, you need already 5 years before you understand actually what's happening in the website. So there you really should hire a specialist to dive through all these hurdles. Well, the project consists of 2 stages. The first part was collect biopsies from patients with ultra-rare mutations. And we collected these biopsies in over 500 patients. And the second stage of the project is that we were testing the compounds of the partners, the biotech partners that were in the consortium. And we had the aim to select the high responding patients and to bring them in a clinical trial with these compounds. Well, this asks for a rectal biopsy because you need some rectal tissue, and it looks a bit weird, but many patients had no problem to do so. And it's not a painful procedure. It's a little bit weird, but it's just like taking rectal temperature, and you have a small piece of tissue just as big as a pinhead, and you can bring it to the laboratory. And as you see here on the top, from these rectal biopsies, you can isolate stem cells from the crypt of the intestinal villi, and you can culture them into balloon-like structures that we call organoids. And you can culture them again and again, and you can passage them, and you can store them in the biobank, and you can use them for assays. And the assay that we developed in the lab of Jeff Beekman is that we can stimulate them with forskolin, and then the CFTR is activated and the balloons start to swell. So the swelling of the organoids is a measure of the CFTR function. And as you can see on the green pictures on the right bottom, is that in a patient with F508del homozygous patient, the balloons are very small, and if you stimulate them with a combination of CFTR modulators, then you see that they start to swell again, so that there's a measure of restoring of the CFTR function. So you can quantify CFTR function, and you can compare a drug efficacy in this model. So it's a rather unique model, I think, because it's a stem cell culture model. It's very patient-specific, so we had for each and every patient has his own model with his own genetic environment. You only have to take a single sample, and you can culture it on, and you can store it for months and years in the freezer, and you can defreeze it and use it again. So it can also be stored in the biobank and be used for further testing in the future when new products come available. And it's also nice that it's a functional assay and it's fully CFTR dependent. So it's a combination of characteristics that make the model very attractive. And what we have published in the meantime during the project is that there is a nice correlation between the clinical response to drugs in vivo and the response that we measure in the organoids. And what you see here on let's see whether it works. That's not the right one. Only this one. Okay. What you see here is the organoid swelling on the x-axis, and this is the clinical response of the patients after treatment with the drug that was tested in the organoid model. And here you see the change in FEV1, and on the right side you see the organoid swelling on the x-axis and the decrease in sweat chloride on the y-axis, and you see nice correlations between the in vivo and in vitro responses. And if you calculate the predictive capacity of the model, then you come up to a predictive capacity of up to 85%. So that's reasonably well. So in the consortium, we started collecting the organoids. So patients were recruited in the individual centers. Each center could look into its own database and see, well, what are the ultra-rare variant patients, and invite them for taking biopsies. And in the end, we had organoids from 47 participating CF centers coming from 16 different countries. And as you can imagine, and as Damien addressed, it is really a lot of work, because we have to translate all the ICFs, informed consent paperwork and so on, all the local medical ethical committees to go into this study. But we managed, and over 2 years of time, or 1.5 years, we collected over 500 patients, and you see here in this graph how they are divided over the different countries. There's a large chunk of patients from Italy, from France, from Belgium. In Netherlands, we helped to recruit patients from Germany, so they were biopsied in the Netherlands. But from many different countries, organoids are available. This is how the genotypes look like that are in the population. So there are some 13% of patients who have 2 stop codons. There are 31% of patients who have only 1 stop codon and another mutation, and the remaining patients have no stop codons at all, but they have all ultra-rare mutations. And well, we started to screen the organoids with the compounds of proteostasis, and they have corrector and a potentiator combination, and also an amplifier combination. But these are the results, for example, of the corrector potentiator combination. And what you see here on the left side is the AUC, so that's the response in the organoids. So you can have a high response or you can have a 0 response. And over the x-axis are the 500 patients. And you see that there are patients who are really highly responsive on the left side of the waterfall plot, but there is also a big proportion of patients that show no in vitro response at all. So overall, we saw that about 30 to 40% of this population of ultra-rare variant patients were responsive to the CFTR modulator combination. What you see here is a reference line that was from delta F patients who were treated with Tez-Iva, so as a sort of reference. So you can't compare them 1-to-1, but you see that it's really for a proportion of patients that this combination of drugs would be a solution, maybe. What we also found is that there is a lot of heterogeneity. And here you see, for example, the swelling in the organoids of patients treated with the PTI combo, and here you see the Vertex combo. And then you see that there are patients who are responsive to the PTI combo but have no response to Vertex or only low responses. And you see that there are also patients who have the results the other way around, with high responses to the Vertex combination and no response to the PTI combination. And there are also patients who are in the middle and they respond to both. But this is a suggestion that it would be good to go for a personalized approach, because not all patients react in the same way to these two different combinations of CFTR modulators. Well, the next step is that we wanted to select the highest responder to the PTI combos, or at that time already the Fair Therapeutics combo, in a clinical trial. And we will run the trial within the network. We also aimed, and we tested the ELOX compound, and we also aimed to do the clinical trial, but in the meantime, the phase 2 clinical data from ELOX became available, as you have seen in the literature and in the news papers, and that trial was not successful. So it's still in the open whether we can do a clinical trial with the ELOX compounds in the future, and whether they will further develop. So this is the trial design of the trial that will be done with the PTI combo. It will be a multi-center randomized double-blind placebo-controlled crossover study. We are dealing with small populations, so that's the reason to choose for a crossover design so that every patient can be compared with his own results. And what we have also done is that we have looked into the time points of the measurement. So we are the statistic analysis will be focusing on mean values of endpoints measured over time during the end of the trial, just to increase the power of the study. There will be a group of patients who are the high responding patients, so just to show the efficacy of the compounds in this group. And we also have selected randomly a group of patients that will go in a parallel trial with the same design, just to compare whether pre-selected patients have better responses than randomly selected patients or non-selected patients, just to show that the organoid model is helpful. But also, when you have the randomly selected patients, you will have a range of organoid response in your group, so then we can say something about cut-off levels in the future if the drugs turn out to be effective. And this was made in close discussion with EMA and regulatory authorities in several countries. So in the end, what will the HCF project deliver to the community? Because that was really the starting point for us as an investigator-initiated study, not to earn lots of money, but to help patients and to move the field forward. Well, the project will deliver, the organoid technology is developed, it's validated. We are currently in discussion with EMA for a qualification opinion, and we are almost to the end of that procedure. Then we have functional measurements of CFTR in the intestinal organoids, and in the meantime we develop the model further so that we can also apply it in airway organoids and in airway cells, both nasal and bronchial cells, just to develop more technology that can be used by the field and other partners. The HCF project also delivers an extensive biobank to the community. So we have not only these intestinal organoids in more than 500 patients in the ultra-rare variant patients, but we also have intestinal organoids collected already in over 700 patients with more common variants, so they are available. And we are also collecting currently airway organoids, and we have already more than 500 patients of all types of variants in the biobank. And the biobank will be used for HCF, for testing the compounds, but it's also open and useful for further scientific work and collaborations with researchers from all over the globe. And we can think about connection with clinical data. We are in discussion with the patient registry to add the organoid results to the patient registry for use for the open community. And of course, the biobank can also be helpful to speed up the drug development programs of current and new upcoming pharmaceutical partners if they have a need for ultra-rare variant tissues. And the third thing, and maybe this is one of the most important ones, also in view of what's told in the conference today, is that we have collected a unique patient population. Because this population will never benefit from the CFTR modulators. We know that they will not respond, and they will know themselves that they will not respond now and in the future. And they have an urgent medical need for treatment. And what's very special is that we have already written informed consent of these patients to approach them for clinical trial, and they expressed one by one that they are interested to participate in clinical trial. So that can be helpful if you want to start up a clinical trial and to speed up the start-up process with these patients, because they are already known, so we don't have to start a study and then start searching for patients, because we can select them up front. So the recruitment period can be very short in this type of approach. Patients are eagerly looking forward to new trials and treatment options, and also the caregivers. I was approached by many of my colleagues during this conference, when are we going to start, because my patients are in urgent need, and I can assure you that we also get emails and telephone calls from patients that are looking forward to what's happening now. Well, for all these patients, the personal-derived cell models are available, and the studies can be done in, well, the well-equipped CF centers that are all members of the ECFS clinical trial network, and all these trials are done in close collaboration within the trial network. And as you can see here on this slide, here are the 500 patients. We will, and these are the responses in the PTI to the PTI combos. Well, we are now selecting the upper group of responses for the FAIR Therapeutics trial, but there are, well, 300 to 400 patients available that will never respond to CFTR modulating therapy, and I think that is a very interesting and helpful population for companies who are working on read-through agents, mRNA, tRNA, or antisense oligonucleotides, or gene therapy. And I think that well, we offer this to collaborate on it, just to speed up the processes in benefit of the patients. Well, I would like to thank, of course, the big group of people who are working on it, and especially I would like to mention Marlou, who is really working quite hard on this project, and she is instrumental in many of the issues around this project. But many people are working on it, and it's good to know that we have a website, and you can read it in almost all languages in Europe. So if you have any questions, you can approach us via this platform and read what it's all about. Thank you.
Are there questions for CORES? Dr. Sermet.
Very much for this very interesting presentation. I was very interested by the pose and F-Kaftor and all the proteostatic combo. So you just presented a comparison with the TESA-EVA effect. So what about the triple combination in comparison?
Yeah. Well, during in the period of the testing, and the testing of the big group, the ETI combo was not yet available, so we tested all these patients with the TES-IVA combo. So that's the most direct comparison. And well, we are now looking into the ETI, but we haven't tested all these patients.
But you have already some results, right?
Yeah.
Okay.
Yeah.
And
yeah, well, what you expect is that the ETI combo is more effective than the TI combo, and you can expect what you see then with the PTI combo. And what we showed here is also the double combo of the PTI. But if you compare the triple combo with the triple combo of PTI with the triple combo of Vertex, you see more or less the same well, heterogeneity. So there are some patients who respond better to the ETI, and there are some patients who respond better to the proteostasis triple.
And I was wondering, considering this variability, did you retest the organoids of patients, or did you resample the patients? Because I assume this might be also different according to the number of, the heterogeneity, the cellular heterogeneity of the organoids itself.
Yeah, well, a part of the project was just to validate the organoid model and to look into the repeatability of testing. So what we have done is repeated testing on the same organoids in the same lab. We have comparisons between the different labs. What we didn't do for ethical reasons is that we re-biopsied patients. It would be nice, but we haven't done it so far, and we don't expect that it will change over time that much because we are focusing on CFTR function, and it will not change over time. I guess if you look to, for example, results of sweat testing over aging.
Thank you very much.
Yeah,
I think
that's great.
we are a tiny bit tight for time, so we'll cut it off at one question. But thank you so much for the presentation.
It's been exciting to see the development of the HIT-CF program over the years, and I am so impressed with the resilience and determination of Kåre and his team to get to this point. So it's been very impressive.
And it's my pleasure now to introduce Anne Stevenson from St. Michael's Hospital, University of Toronto in Toronto, Canada. Anne's going to talk about harmonization of registries on a global scale.
Thanks very much for the invitation to speak today. And I'm going to just launch right in as soon as am I using this? Okay. I have no disclosures. So I'm going to talk first a little bit about the history of the registry. People are very familiar with the registry, but not necessarily how it was developed, which is an interesting story. And challenges that we have when comparing registry data, because there's a lot of data out there, but how can we compare them in a reasonable way? And is this even doable? And end with some of the future directions related to comparisons. So the US CF registry actually has been around for quite some time, and it started because there was a pediatrician in Cleveland in 1957 who was saying that the mortality at his clinic was actually quite low compared to what the estimate was nationally. And there was no database, no one really knew what the national mortality was. But he kept on making these claims, and finally the US CF Foundation thought, you know what, I think we should give some money to Dr. Warren Warwick and collect some statistics from the clinics across the country. And so he did that, went around to 31 different clinics, and did a report at the end, which in fact did show that the median survival was 7 times higher at Dr. Matthews' clinic compared to the other sites in the States. And this was sort of the first time that people realized, you know what, we need data to actually support our claims, and that's much more powerful than just sort of waving your hands and saying my clinic is really great. The US CF registry was sort of developed, and what people often don't realize is it actually was a joint registry with Canada for many years in the beginning times. The USCF Foundation came to Cystic Fibrosis Canada at the time and said, would you like to contribute data down to the registry? Many of the Canadian clinics said, yes, that sounds like a fantastic idea. So they sent their data down to the US. And this is one of the first joint reports that were published in 1976. And Dr. Mary Corey, who's a statistician in Canada, she actually gave me these reports. I have them in my office because she was around when it was all started. And I look at these reports and it's fascinating just to see, you know, they were made on a typewriter, which my children don't even know what that is. And just seeing the different graphics and the tables that were made in these early days is really fascinating. So this is just a table from that report showing that there were just over 2,000 Canadians with cystic fibrosis from 1967 to 1976. And in the States, there was about 10 times that number, which is still the case. Theirs was from 1966 to 1976. Now, a lot has changed since the 1960s. Not only the fashion, but actually the registries are now completely independent. And in 1980, the Cystic Fibrosis Foundation in Canada said, you know, I think we should take our data back and have our own registry separate from the US, which is still the case. And also, you know, we've had to get consent from patients and we've had to get REB approval. So it's much different than the early days of the registry. But many countries actually have registries and they produce glossy reports with nice pictures and beautiful graphs that are quite different from the early days. And this is slightly different than what we've heard so far, where these are projects you've heard about that were developed with a specific question in mind and collecting data prospectively. The registries obviously are retrospective. We're sort of limited to what's captured in the registry when we use these data. So how can we, you know, use the data that's published and available and compare between countries? And why would we even want to do that? What's the purpose of doing that? And it's to facilitate comparisons might be important to identify perhaps gaps in care. So then you can go back and figure out why there was a gap in care, and that might help advance CF care globally. Also, it might be important to increase generalizability of findings. So there may be somebody who develops a predictive model, for example, with one registry in one country, and they want to see, does it apply if you put it into another data source, another country, and you need to have the same variables in order to do that validation study. And furthermore, it increases our understanding of things like rare outcomes that may be not possible to do in one country or with one registry, or there may be natural experiments that occur that you think, okay, I'm going to look at that because I know in one country they have access to this sort of environment and another country has a different environment. And so let's look at the differences between them. So within these registries, we know that there are important outcomes like lung function, nutritional markers, vital statistics. All of those registries that I showed you up on that slide pretty much capture these data. So why can't we just compare the registry reports to say, this is how one country does it, and this is the results from another country? The problem is that sometimes these variables are displayed in a different way, and also the actual definitions of these variables are slightly different, and I'll show you what I mean. So this is lung function, which is shown in two different reports, and you can see that the first thing is that there's different lung function categories that are used for the two different reports, so it's hard to do a direct comparison. Furthermore, there are different age categories, so you can't sort of look at one particular age and say, what's the lung function like in country A versus country B? Furthermore, you can see from these graphs that it's very difficult to even determine what the lung function is exactly based on how the graphs are displayed. So you really need to get into the raw data to do that comparison and make sure that the data is harmonized so that you can make an appropriate comparison. And I say that because you don't know from the report often, are they using the best lung function of the year? Are they using the average lung function of the year? And if I was going to just do a simplistic comparison, I might say, well, this country A has better lung function than country B. But if they use the best lung function of the year, of course they're going to have better lung function. If they didn't, that would be kind of strange. So you really need to look at how the data are collected and reduce it to the lowest common denominator in order to make an appropriate and fair comparison when you're doing comparisons between countries. Now, what about survival? Clearly, that is not a subjective outcome. Someone is alive or dead. That surely should be something we could look at these reports and do comparisons. Unfortunately, it's not even that simple when you're talking about survival. So on the top left-hand graph, you can see what happens in this case. What we did in a simulation study was we made it so that there was an increasing proportion of missing death dates in the registry data. This was using the Canadian registry. And we calculated the median age of survival. So as you would expect, as you're missing more deaths, like say your follow-up is not great, but you don't know if someone's dead and you think that they are still alive, you're overestimating the median age of survival. Similarly, if you're lost to follow-up, if your registry has a lot of people that are lost to follow-up, as shown on the bottom left-hand side, the median survival would be underestimated. Now, in reality, these happen sort of together. You know, you might be missing a few deaths and you might have people lost to follow-up. So maybe the ultimate median age of survival is very accurate, but you don't know exactly until you start looking at the registry how complete it is, what the impact of that bias might be. Similarly, often it's not explicitly stated, or at least previously it wasn't, it's a bit better now, but wasn't explicitly stated whether you were including transplanted patients. So if someone had a lung transplant and you did survival analyses and included those deaths after transplant, the median survival would look very different compared to censoring at transplant. And that's shown on the right-hand graph where you see a survival graph, and in the dark line, that's when we censored at transplant, and the light gray line was the exact same data set, but we included transplanted patients. So you'd see that the median survival would be lower in the group that had the transplanted patients. So you need to be careful when you're reading these reports and doing these comparisons that you're actually comparing apples to apples when you're doing this. Now, these differences exist, but I would say that it is possible to successfully harmonize data in order to answer important questions. You just need an appreciation of how the data are collected and a unified approach. And these are just some publications that have been published using comparisons, using registry data, comparing between different countries. And that's where you sort of spend some time finding out what the raw data is and coming to an agreement about harmonizing those variables to bring them to the lowest common denominator. So I think this is a bit of a change of the way that we were thinking, because there's this global harmonization, or what we now say the CF Global Registries Group. We initially thought, okay, we should harmonize the registry to collect the same thing and define things in the same way. But the reality is that's very difficult, even for things that are very simple, like age at diagnosis. Some people would say, well, when is that? Is that when you get the genetics back? Is that when you get a sweat test back? And it's sort of just, you go down these rabbit holes, and in the end, you can't make any decisions. So I think we sort of changed our way of thinking to think that's unrealistic, because one size doesn't fit all. And you have to think about the different registries that exist. There are some, like I told you, the Canadian and the US registry that have been around for decades. There are others that are in their infancy. And the questions that can be asked of those registries are very different. If you have an established registry, you might want to look at the nuances of certain treatments and what the impact of those treatments might be. If you have a very new registry, you might just want to know, how many patients do we have in this country? What's the lung function like of these patients? So you're not sort of thinking in the same way. You have to think about what's the purpose of the data collection and the importance of outcomes. So this graphic I like because it shows all the different registries. We can communicate with one another and talk about how we define variables. But when we have a specific question in mind, we can actually bring them together to harmonize them and overlap them so that we can answer questions. So how do we do that? We really start with asking, first of all, who is in your registry? If you have a broad spectrum of disease within your registry, then you need to be aware of that, because if you're comparing a country that only collects data on people with severe CF, it's again not going to be a fair comparison. Are there a lot of people who refuse to participate in the registry in your population? Are you including post-transplant patients? Because a lot of times those people are lost to follow-up and you don't get a lot of outcome measures after transplant for certain registries. So this helps you to assess the potential bias, and then you can develop a clear question, and in collaboration with another registry team or registries, you can then define those variables in order to ask and answer your question. It's important for registries to have a clear instruction manual which defines variables within that registry and have a standard process and approach to this, and that this is transparent. So back in the early days, the Canadian registry really didn't have a lot of good documentation, and we've corrected that. So now when we try to do a collaboration with someone else, we can say, okay, we define this complication in this way. How do you define that complication? Or we're collecting this first stable measurement of the year in terms of lung function. what are you collecting? And it's much easier to then see how you can harmonize the variables. But these documents are kind of changing over time, and they are evolving, so it's important to just make sure you're looking at the most complete and up-to-date one. And this is exemplified in this CF Registry Global Collaboration, which I'll just use as an example. There's many examples of doing these international collaborations, as we've heard about already in this session. And we've met long before the COVID pandemic. This group has met together to try and define these variables. But the COVID pandemic really kind of made a pressing issue that we needed to address quickly. And we started with just getting together with 8 countries to sort of say, what is the impact of the COVID-19 infection on patients with CF? And we need to find this out fairly quickly. And that allowed us to find out that, reassuringly, patients with CF, if they got infected, generally they tolerated it better than I think the community thought they would at the very beginning. This collaboration has expanded to 22 countries and now 61 countries that get together to talk about, right now we're focused on the COVID-19 pandemic and still have some questions that we're trying to answer with that. But we're going to hopefully have additional research questions that we will have in the future. And so I think this has proven that this is a viable approach. This is one of our Zoom calls that we had. And we've been fairly productive in publishing several papers looking at this, the impact of COVID on CF. We were successful in applying for grant funding through the Canadian Institutes for Health Research to support this work, and in particular to support data collection in low to middle income countries that didn't necessarily have an expansive infrastructure for registries. And we hope that the next paper that we will be looking at the impact of COVID-19 on the trajectory of lung function over time will have over 3,000 cases and have, as I said, almost 60 countries involved. So this is a continued collaboration beyond COVID, and we will be addressing other questions, perhaps looking at rare mutations or disparities in care, et cetera. And so our vision for the future for this collaboration really is that, there's a lot of diversity and experience in this group, so it's sort of a springboard for ideas and projects, almost like a matchmaking. We can bring people together to answer questions with the appropriate groups. Not everybody has to be involved in every single project, and provide advice, mentorship, and more importantly, advocacy for the CF group, because I think when you come together as a larger collaboration, you have a more powerful ability to advocate for your patients. Now, this is talking a little bit about balancing needs versus wants, which doesn't have so much to do with harmonization, but I know that it's always difficult to know how much data do you collect in your registry, and do you want to minimize data collection so you get very good, robust data versus maximizing data so that you can ask more granular questions. And I think it really depends on the country, it depends on the registry, and what the purpose is, the infrastructure, and the support that's available to them. In terms of future directions, I think that we have to consider how we can leverage registries by linking to pre-existing data sources. So in Canada, we are linking the registry to the health administrative databases within the province of Ontario, and maybe expanding it to other provinces where there's a lot more granular data about healthcare utilization. And so we're not asking the CF clinics to import that data. We know it exists somewhere else. How can we bring those two data sources together? We are considering, patient-reported outcomes. That's been discussed a lot over the conference, how we can import that into registries, how we can collect data from wearable devices. And as was mentioned earlier in one of the talks in this session, how can we link back to the registry? So you might have a research question and think, I need to collect information that's not in the registry, and that's fine. But make sure you think, hey, I'll collect the registry ID number as well so that I can go back and I don't have to recollect all the information on lung function, nutrition, microbiology, hospitalizations. That's genotype. All of that is already in there. But then I'm going to just focus on collecting the important information that's not available in the registry. So the take-home messages are really that these are valuable resources. They need to be maximized and leveraged, and I think you can see through the conference over the years, more and more research is done with these registries. There are data from multiple registries that can be used and combined and compared. It's just that you need a thoughtful kind of unified approach in order to do this. Think about primary data collection linking back to the registries. And this global collaboration, I think, is a really great opportunity to share, collaborate, advise, and mentor people. And I want to acknowledge the group that is involved in this. Obviously, it's a very large group of people. These are the 61 countries that are involved, and this was the picture of the European CF meeting in 2022 with the group that was attending at that time. So if anyone's interested in hearing more about that, feel free to email me. I'd be happy to connect with you to see if there's any opportunities that you can do using the registry. Thank you very much.
And thank you so much. That was a wonderful overview. And so to me, one of the questions is, can an investigator just send you an email and say, can you give me an anonymized data set of all of the registry information you have? I imagine there's a process.
There's a process, yeah.
And there's a
You
cost.
can't, and I'm not the gatekeeper for all of the registries. I am the medical director for the Canadian registry. But each registry has its process of accessing data. So there's an application process that you would go to talk to the individual registries. If there was a question from a more global scale, I think maybe reaching out, and certainly you can email me to connect with the global collaboration. That's a different question. And you know, each individual country has their processes in place for collecting data for registries, and we've been able to facilitate without, you know, crazy contracts, because that is totally scary. Connect everybody together with de-identified or anonymized data sometimes to collaborate and bring that data together. So there's two opportunities depending on which data you want to use.
Are there other questions? We have a microphone that's working.
Thanks very much. You're okay.
Okay. All right. Last presenter of the conference, the enviable or not enviable position. But I'd like to introduce Dr. Isabelle Sermet from Necker Enfants Malades in Paris, and who is also, as Dr. Downey mentioned earlier, the chair of the standardization committee for the CTN. So she's going to talk today about global standardization of biomarkers, including spike chloride, sputum biomarkers, LCI, and imaging. It's a big task for the last talk of the conference. Thank you, and I'll let you go for it.
Thank you.
So, thank you everyone to be there still at the last presentation of the conference. So I'm going to try to speak about the global standardization of biomarkers and how we really need to be together to do this task. So I have no disclosure to present, but first of all, why is this global standardization so important? And as you can see here in this map, which displays the estimated number of CF patients worldwide, more than 160,000 patients are estimated, whereas only 100,000 are diagnosed. Moreover, more than 80,000 should have access to highly effective modulators, but only now less than 20,000 are treated. So this indeed shows the potential to improve the rates of diagnosis and of treatment of the patients. And it is our duty for all of us in this room, for all of us attending at this conference, for all of us taking care of CF patients, to improve diagnosis, to improve their prognosis, and to make them access to new therapies. And this begins by global standards of care access of all the patients who have this right. And the first step of that is global standardization of biomarkers. First of all, let me remind what is a biomarker. It is a characteristic that is objectively measured and that can be evaluated as an indicator of a normal biologic process, so a diagnosis biomarker, of a pathogenic process evaluating the natural history of the disease, so prognosis biomarker, but also of a pharmacologic response to a therapeutic intervention, a safety, or a predictive biomarker. So biomarkers are outcome measures that can be evaluated either in a clinical setting or in a research setting. So why do we need global biomarker standardization in cystic fibrosis? First of all, and above all, because we have small population of patients at each medical centers, and this requires indeed standardization of the outcome measures which are specific to cystic fibrosis. Because also we need to better phenotype the patients. We need to fuel the patient registry with reliable data. And we also need to identify unique target populations for clinical trials or investigator-initiated trials. But above all, because we need to accelerate drug development and CF-related research, and if we have standardized outcome, this will enable generability of the results because the data collection will be, there will be a uniformity of data collection because of the expertise of the personnel in the centers. And because there will be a decreased variability of the data, this will decrease the number of patients needed per study. And finally, this will also enhance the expertise of the sites and of the people at each site, because this will increase their scientific knowledge and their expertise for each specific markers. So, which are the steps to go forward for a global biomarker standardization? Of course, the first step is implementing standardized operating procedures, but very importantly is also to better characterize the CF outcome measures that we use by assessing their feasibility and what is called a clinometrics, and characterizing them in terms of both sample size and clinical relevance for the patients. The second step is to implement central core facilities to provide training to sites, to have qualified members at each site, to really have a pool of sites available for sponsor, and to also to provide centralized quality control of the data provided by the centers. And finally, also for very specific biomarkers, provide a central overreading system to improve the consistency of the evaluation and of the scoring. But very importantly, this centralization, standardization of biomarkers is enabled, accelerated, I should say, by therapeutic networks. And among those, the therapeutic development networks is really a paradigm for genetic and orphan disease. It was launched in 1998 by several persons in this room, including George, but Bonnie Ramsey and Bob Bell were really visionary people who envisioned the challenges and also the tremendous opportunities of the network, including of the standardization of the outcome measures. And now, as George said, in 2022, GDN is more than 20,000 patients, 91 sites all over the US in 44 states. More than 1,500 patients with CF have been enrolled into studies only this year, and TDN has reported 16 study trial results, so it's really a huge task force. Cystic Fibrosis Canada Accelerating Clinical Trials Can Act has now 10 sites in 2021, which includes 60% of people with cystic fibrosis in Canada and a large number of clinical trial participants. In Australia, there is not an established network, but a lot of centers include also a lot of patients in clinical trials. And in Europe, we launched this clinical trial network in 2008, thanks to a Eurocare CF EU European Union program. And at the beginning, as you see, it was 18 sites in 8 countries, UK, France, Italy, Germany, Netherlands, Denmark, Ireland. Then the second wave of expansion in 2011 enlarged to 30 sites in 11 countries, with Spain, Sweden, and Portugal. In 2015, 43 sites in 15 countries, Czech Republic, Poland, Switzerland, and Israel. And finally, the last wave was in 2019, where we reached 58 sites in 17 countries, including Austria and Hungary. So CTN in 2021 is more than 20,000 patients with cystic fibrosis all over Europe in 58 sites, 17 countries, including Eastern Europe and also Israel. So 41% of patients with cystic fibrosis in Europe and nearly 20% of all patients with CF worldwide. But this is a richness, but this is also a challenge to have and to monitor so many people. Just an example, as you can see here, the median age of death according to the country income, low income, middle income, high income. And you can see the disparity of this median age of survival. And clearly, it improved between 2014 and 2019, but still the differences remain, highlighting again how it is important to access to this patient in low-income countries, to make them access to healthcare and to improve their CF outcomes. This is a richness, and this is also opportunity, as highlighted by the genetic diversity within CTN. Here, the difference between, according to the Delta 508 homozygous status, between the homozygous, heterozygous, and those patients without any Delta 508 genotype. And indeed, Delta 508 is found mainly in north of Europe, also in Croatia and in Denmark, but when you check for G542X, it's found mainly in southern Europe, but also in Armenia, whereas it is very rare in Northern Europe, and 1303K, mainly found in southern Europe, but also, very interestingly, in Iceland, G51D in Ireland and northern of Europe, very rare in southern and eastern Europe, and finally W1282X, very frequent in Ashkenazi in Israel, but also in Georgia. So very important panel of diversity. So how can we make that, how can we reach to this global standardization and being stronger together? I will illustrate that by illustrate the task force between the different networks in US, Canada, Europe, and also centers in Australia by taking examples of relevant biomarkers which actually serve as early signals for therapeutic efficacy in the development of new treatments in cystic fibrosis. First, the sweat test. Since it was discovered and implemented by Paul de Santagnes, who observed the increased level of chloride in sweat of patients, it has been indeed standardized, both sweat collection by pilogel iontophoresis and West Coast collector, and also the chloride measurements, mainly ChloroCheck and chloridometer. But very recently, because of the challenges with the new CFTR modulators, it has proved as a remarkable pharmacological biomarkers, and it is used in proof of concept studies. So it has obviously been one of the first biomarkers which has been standardized within SOP, both at TDN and CTN. And we worked together, CTN and TDN, for a shared SOP in 2018, which modified both SOP, TDN, and CTN, in fact, including new guidance photos, but also really including very important guidelines to how to induce the sweat and to induce and to perform that on the two sides on opposite forearms sequentially and not simultaneously. And also we had guidelines about the minimal volume to be collected and the maximum time to collect the sweat. So recently, this SOP was upgraded and updated because WestCore has now launched the MacroDuct Advance, which includes elliptical collector, which seem to be more optimal to collect sweat, and this is associated with also a specific inducers, and it is all the more important in Europe that because of the new EU regulation, Pilogel has been upgraded to medication status, and WestCore is reducing its portfolio only to MacroDuct Advanced inducer. So TDN and CDN have harmonized the SOP for sweat test. Because we have so many languages. In Europe, we have made a lot of leaflets to explain to patients in multiple European languages and increase accessibility to this test in the different countries of CTN. However, although the sweat test collection is really well standardized, there remains in Europe a lot of inconsistency in performance and reporting of sweat tests. And this is mainly observed in low-income countries and also in countries with low prevalence of cystic fibrosis, mainly because of the cost of the equipment and also of inconsistency in the measurements of the sweat chloride. This is also because we lack external quality control in many countries in Europe, and CTN attempted several years ago to build a core facility, but this fails because of the high complexity of the transporter rules to send biological samples. This is also because we need to better train and better certify the different sites. At TDN, it is really remarkably done with e-training and face-to-face training and certifications for each site. At CTN, we have trainings at ECFS, but this is still on an irregular basis, no certification in place, no certification at CANACT as well, and we are now in the process of harmonizing our process of certification and training with TDN, following the example. The second biomarker is lung clearance index. It is an old multiple breath washout, which has recently been rejuvenated for research in cystic fibrosis. And indeed, it was first published in the 1951 in GCI journal, highlighting the interest of evaluating the ventilation heterogeneity. And since then, it proved in CF able to discriminate between health and disease in preschool children with mild disease, and it's also very much used now in clinical research to detect changes in patients with mild disease treated with modulators. So the example of lung clearance index standardization is absolutely remarkable because both the experts in US, experts in Europe and Australia work together to implement and standardize this evaluation. This is really due to the close collaboration between experienced operators. So in 2013, 2015, there were a lot of discussion for the gas which could be used for this lung clearance index test between nitrogen and SFCs. And finally, there were critical issues with SF6 because of the cost, also the availability, and at this time, it was also considered as a very potent greenhouse gas. So it was decided by the experts, and this is highlighted here in the standard operating procedures, to move for nitrogen and to implement a device-specific SOP, which was done by the experts, approved by the vendor, actually Ecomedics, and adopted both by TDN, CANACT, CTN, and also Australian sites. This has been since then used in many clinical trials by pharma, but still the three core facilities work together to support clinical trials, exchange data, and there was a recent data reanalysis of all the results because of the publication that there was a sensor crosstalk error causing overestimation of lung clearance index. So all the three core facilities have worked together to reconsider all their data. So this is a remarkable example of how the networks can work together. How does it work at the CTN for training and certification process? But it's also the same case in Toronto, which trains TDN sites and CANAC sites. So at CTN, the LCI core facility was launched in 2014, and since then it has trained and certified 46 CTN centers, more than 170 operators. Those certification requirements are non-protocol, non-sponsor specific, with now post-COVID area hybrid training with online modules and face-to-face visit to sites. So 2 operators are trained by sites. And finally, this is a mixture when we consider the operators trained of operations really wanting to be certified for trials, but also for own research, and even now for clinical care of patients. So these certificates are required, of course, prior to performing the testing and transmitted to the company. So this LCI core facility is also central reading and also quality control. So as I said, North America, Australia, and the UK London centers have supported more than 20 clinical trials in this collaborative task force. And now the CTN centers is launching a quality control project, identifying sites with challenges, making site survey to really evaluate the working conditions, and finally have a personalized report for each site with a set of recommendations on how to improve. Third example, lung imaging. Lung imaging can be used as a prognosis biomarker used in care setting, but also as a predictive biomarker, which can be used in research by really aiming to assess the modification, for example, of inflammation at short term induced by CFTR modulators. And I must say that CTN puts a lot of effort into standardization of lung imaging. And this was initially, a few years ago, launched by guidance documents, which was made both by CTN experts, but also CF-TDN experts, Alan Brody, Paul Gilman, and Scott Nagel, which concluded that the general SOP was absolutely impossible because intrinsically lung imaging is really very difficult to standardize because there is a large heterogeneity among vendors, among sites, also continuous technological implementation. So this first manuscript recommended really general guidance, including quality assurance, budgeting, IRB consideration, and very importantly, radiation safety, and how to make CT data acquisition and data analysis. The only SOP that we have at CTN is a spirometry guided chest CT to improve quality acquisition, so pre-analytic as evaluation of the lung imaging itself. So how does it work at CTN? We have launched a lung analysis core facility in Rotterdam, which first is now performing CT training and certification process. As you can see here, it's really an e-training, not a face-to-face training, with webinar, first to discuss the standardization, the importance of lung imaging, and how the sites perform, the candidate sites perform actually in routine, and the bottlenecks that he could endeavor. Then there is an online e-training, several hours, how to perform this CT acquisition, both on the clinical sites, but also on the radiology site. And finally, there is evaluation at the lung analysis core facility of the scan acquisition and the reconstruction parameters. And finally, the certification is granted once the e-training and also a phantom scan is completed. So to date, 25 CTN sites have been certified, 18 are in the certification process, and 8 are scheduled to enter the process. But there was a considerable breakthrough recently by the association of these imaging central reading facilities at CTN in Rotterdam with a Dutch medical company, which is Therona, which is specialized in automated image analysis for artificial intelligence. And indeed now, thanks to this collaboration, the chest CT can be analyzed and standardized analysis longitudinally for clinical data, for example, fuel registry, but also for evaluation, short-term changes, for example, of inflammation of patients treated with CFTR modulators. And this is mainly due to the large amount of work which was done by Harm Tiddens and Australian team as well, by implementing score analysis, very interesting score, which is the Pragma score, and which enables this standardization work. We have launched also at CTN specific imaging special interest group to implement recommendations and guidelines for CT monitoring, to work also on low dose strategy and recommendation, and for MRI protocols. So CT monitoring strategy subgroup aims to have ACFS IRIS guidance on regular monitoring, but also specific situations for lung-tech CT indications, for example, MBPA, NTM patients follow-up, or hemoptysis. Currently, there is a survey among adult and pediatric centers, and with a joint position paper, which will be proposed in the next months, also working on ethical aspects of ionizing radiation, focusing on prudence, accountability, and transparency for the patients. The low-dose chest CT subgroups ask the questions, how low can we go, and should we go low? And it works now on reviewing the current thoracic CT protocols which are in use in the different centers, which are the typical radiation doses the patients with CF are exposed to in the CTN centers, the dose reduction strategies which are employed, the adherence with those reference limits, and finally, assessing the balance between the image quality and the radiation exposure. MRI subgroups aims at standardized MRI protocols to define a set of sequence for chest MRI, but also to summarize the current chest MRI practice in the different centers in Europe and define the current indications for MRI in cystic fibrosis. Finally, last but not the least, sputum biomarkers. They can be considered as routine monitoring biomarkers to better characterize the patient, but also as research biomarker. So again, CTN has aimed to standardize biomarkers, sputum biomarkers, really to improve the standards of care, implementing different SOP for frequency and methods of respiratory sampling, isolation, identification of Pseudomonas aeruginosa, and also proposing definition about Pseudomonas aeruginosa status in patients, new infection, eradication, what is a Pseudomonas chronic colonization. And importantly, it has put the way to sample the patients at the core of this SOP. And as you can see here, in non-sputum procedures, we recommend sputum induction, and we have put a lot of effort to update our old SOP because now patients are less and less sputum producers. So we have updated our existing SOP. We have updated that according to the age, below 6 years, above 6 years, with a specific flow chart for sputum induction procedures, with infographic for patients above 6 years, how to collect samples for microbiology, routine care, but also for research, and also guidance for physiotherapists or nurses, also for patients below 6 years, for routine, but also for samples which could be used for research. We are now finishing a video which will be translated in many languages in Europe, really to guide sites to perform sputum induces, not only in adults and in adolescents, but also in preschool children and in babies. Again, these sputum induction procedures are translated in multi-European languages to reach maximum of patients and of sites. But finally, most importantly, sputum biomarkers can be used also for research, and they are increasingly being used for research. CTN has published this year a paper assessing the potential of inflammation biomarkers in sputum, but evaluation in Europe and within CTN is based on research laboratories, whereas in TDN, very importantly, there is a central laboratory able to perform and to analyze all these inflammation biomarkers, not only inflammation biomarkers, but also other biomarkers that could be very importantly assessed in the sputum, such as microbiota, rheology, mucin contents, and also, importantly, mucociliary clearance. So these different labs should share expertise and samples between networks. So to conclude, biomarker standardization in CF worldwide has reached considerable achievements, but there is still much to do. Very successful trans-network global standardization is illustrated by the lung clearance index, also for sweatless, but we lack quality control. Lung imaging requires the same workflow, and for sputum, we really need, for this very valuable and non-invasive biofluid, we really need to work together to validate new research biomarkers, because at the final end, these relevant biomarkers can be used in the setting of the CFTR modulators, but also beyond, so that treatment burden can be minimized in patients, and we could distinguish those at high risk of lung infection and those who could have a minimal treatment burden. And with this, I just want to finish by acknowledging all the people at CTN, so including Damien and Kate who is there. We are working all together, but also at TDN, Jill and George, who really very kindly also shared information for this presentation, and also Brad Kahn from Canact. Thank you very much.
Isabel, that was absolutely wonderful overview of that absolutely critical area for the networks and for future drug development. It seems that for some of these biomarkers, it's important to have one machine and have that machine under the control of the research team, as opposed to having to work with the radiologists and different equipment. And it's just a very striking lesson. Of course, the imaging is far more complex than it is to gather sweat. Do you think that it sometimes is easier to send a patient to provide a sample than it is to send a sample across countries? Maybe a question for Damien and Court.
Well, I think it's more simple, theoretically more simple, to send the sample. But in fact, yes, we have a very complex EU rules, but maybe Damien has better understood with the core CF project. And we were very surprised when we wanted to implement this core facility for sweat, for quality control, just to implement quality control. We were so surprised that it was so complex. But I still keep hoping that we could be able to do not only within Europe, but also trans-network between Australia, between TDN, between Canada and Europe, to send samples.
And do you think there would be agreements that could be set up for any sample that would be uniform regardless of the protocol?
Definitely doable. So we do hope. We do hope. We need to work on that.
We can end on definitely doable. I think that's a fantastic way to end. Thank you everyone for attending. Hope you enjoyed the conference.
Yes, and thank you to our speakers for providing us this update across the globe.